Patient adherence is very challenging, and drug metabolism can vary person to person. So objective measurement of drug levels may prove to be important in preventing the emergence of resistance during TB treatment.
If adherence could be more accurately and easily quantified, then healthcare providers could develop a treatment plan that would limit the development of drug resistance.
We have developed the first multiple analyte mass spectrometry panel for multiple TB medications within small hair samples in a single assay. Our multi-drug panel only requires 1 2 milligram sample per patient and uses neither solid phase extraction or filtering step to prevent any loss of sample volume.
Objective measures of drug exposure measured early in the treatment course could potentially alert clinicians to problems with individual drugs in the treatment regimen.
Hair samples must be pulverized on the day of analysis for an accurate drug concentration level measurement.
To acquire hair samples, after obtaining written informed consent, get materials ready, including a thin label, 2 participant labels, a pair of scissors, an unfolded piece of aluminum foil, an alcohol wipe, and a plastic bag. Wipe the blades of the scissors clean with an alcohol wipe. Go to the back of the head and lift the top part of the hair using a hair clip if desired. Gather 30 strands of hair from the back of the head and isolate the thatch with your hand. Take the scissors down to the scalp and cut the thatch of hair.